Restriction enzymes are essential tools in molecular biology and forensic science. They are enzymes that cut DNA at specific sequences, producing fragments of varying sizes. In nature, restriction enzymes are used by bacteria as a defense against viruses. When a virus infects its host, it injects its DNA or RNA with the goal of hijacking the host to reproduce. What the bacteria does is label its own DNA by methylation. What this does is make the methylated DNA recognizable to the restriction enzyme. But since the viral genetic material is not methylated, the enzyme will recognize it as foreign material and destroy it. There are 4 different types of restriction enzymes. Type 1 restriction enzymes cut DNA at random, far from recognition sequences, hence why they hold little practical value, due to the fact that they do not produce discrete restriction fragments or distinct gel banding patterns. Type 2 restriction enzymes, however, generally do cut at or near the recognition sequences and create discrete restriction fragments and distinct gel banding patterns, which is why they are used for DNA analysis or cloning. Type 3 restriction enzymes cleave outside the recognition site and require 2 oppositely oriented recognition sequences on the same DNA molecule. Unfortunately, these rarely give complete digest. And finally, type 4 restriction enzymes. Ironically, these enzymes look for methylated DNA. Examples include MCR, BC, or MRR from E. coli. They serve as a defense against foreign methylated DNA. In electrophoresis, restriction enzymes are essential as they are used to cleave the DNA into various pieces, which are then loaded into the gel. Since the longer DNA fragments are heavier, they will take longer to travel across the gel compared to the shorter DNA fragments, which are lighter and take less time to do so. Bringing us back to our first example of restriction enzymes. They can also be used to make bacteria synthesize human insulin. Using restriction enzymes to cleave the bacterial DNA, we can insert the human insulin gene, and after, the 2 parts will reunite and the bacteria will begin to synthesize insulin. This will allow us to have a lot of insulin made at a cheap and quick pace, helping treat diseases such as diabetes for millions of people worldwide.